So here's one of the amazing things that happens. Put this down. I want you to look at the color inside this endosome. So remember, here's the cell membrane, and you have to sort of imagining it continuing here so that there's a little bubble of cell membrane. So we call that an endosome. And you notice how David has now colored all of the proteins that are in this endosome red. And that's a key to trigger, to make you think of the fact, emphasize the fact that we now acidify the endosome. It's another phrase, couple receptor-mediated endocytosis followed by acidification of the endosome. What does that mean? It means we're going to drop the pH in which this virus is in from pH 7 before the endocytosis occurred. Now it's going to drop to pH 5.5. It's like 100-fold more protons inside. So a dramatically different environment. How does that happen? Well, there's some little proteins here labeled V. This is a vacuolar ATPase. It's a proton pump. It's just pumping protons into this relatively small endosomal space. So the pH drops. And now, so that's the key to this. There's a pH-induced conformational change in this protein right here. So remember, if we're looking at this right here, this protein, one subunit of this protein, has to get out of the way. So this protein, the hemagglutinin protein, I want you to look at the shape of that protein on this virus. And most of the shapes of these proteins are the same shape that it had at pH 7. Okay? At pH 7, remember back there in this panel, we had the hemagglutinin protein bound to the receptor. And all of these have the same shape. Inside this endosome, which is being acidified, we now have a situation in which about 50% of them are the same, but the other 50% are dramatically different. You see 50% of them have this three subunits, some tails sticking way up. And then if you look further, here's one with the 3 tails, and they're now embedded in the cellular membrane. Another one here. So what is that? These proteins have undergone a pH-induced conformational change. You've changed their shape because they're now in this acidic environment. These other hemagglutinin proteins will very soon pop and adopt this conformation as well. But this is just, David's captured this in the process of happening. Okay, so with this model, I can explain what that conformational change is. In this protein, as well as the spike protein of coronavirus, there's a part of this protein which is called the fusion peptide. And it's a short sequence of amino acids, maybe 10 or 11 amino acids long, and they're all hydrophobic. And why is that important? Well, hydrophobic peptides like to bury themselves in hydrophobic things. And perhaps you already know that the membrane is hydrophobic. So this little piece of the protein would very much like to be buried in a membrane. But in the initial structure of this protein, it's way down here near the viral membrane. What we want to do is to get this fusion peptide, we want to get it way up here where it can harpoon the cell membrane. So how does that happen? Well, here's the conformational change that happens. You see this blue loop of protein structure here? That actually coils up into an alpha helix, which has the effect of lifting this fusion peptide from way down here to way up here, where it can now harpoon, embed itself in the cell membrane up here. And that's happening not just once, but with the other 2 subunits, which is why in David's figure you see these 3 fingers or arms extending up, having buried their fusion peptides into this cell membrane. So now you'll notice that these hemagglutinins are no longer bound to a receptor protein using their receptor binding domains. Instead, the protein has harpooned the cell membrane with these fusion peptides. Okay, so now the next thing that has to happen is that the receptor binding domains have to get out of the way so that these two membranes can come closer together. And I want you to pay particular attention to the distance between this end of the protein embedded in the viral membrane and these three fusion peptides up here. They're pretty far apart here. Now we move to the last panel, in which David shows a further conformational change. So on either side of this, you can now see that the viral membrane here has fused with the cell membrane here, and the viral RNA is now moving into the cytosol. Okay, look how close this end of the viral membrane, is the hemagglutinin protein buried in the viral membrane, is now very close to the fusion peptides that were embedded into the cell membrane. And that has happened because all three of these receptor binding domains have sort of fallen away. Don't want to get into too much detail here, but there's actually something called a coiled coil. These are just alpha helices of those three different subunits that are just wrapped around one another. And as they wrap around one another, they bring these two membranes closer and closer together. When they get close enough together, they're just going to spontaneously fuse and create this opening that the RNA goes through. What do I mean when they spontaneously fuse? So if you've ever fixed spaghetti, you may have a pan of water on the stove, and you put some oil in there. And you might get several droplets of oil that are floating on the surface. They don't mix with the water because they're hydrophobic. But if you watch, a couple of those droplets will move around, and whenever they happen to touch one another, they will automatically just fuse together into one larger droplet. So that's the same sort of thing that's happening here. You've got a viral membrane here and a cell membrane here. And if you can bring those two close enough together, they'll just pop together, and the RNA is now free to move into the cytosol. So that's the infection process.
Hi there, Mark here, a colleague of Dr. Tim Herman's. And if you enjoyed this video, make sure to check out our other coronavirus resources available at www.3dmoleculardesigns.com/scienceofcoronaviruses.htm, including a paper modeling activity where you can create your own physical model of a coronavirus. We hope you enjoy, and thanks.